Hello to everyone that has found themselves here today. So today we're going to talk about why memory isn't stored in one place in the brain. For decades, scientists believed that memory loss, especially in Alzheimer's disease, was caused by one main culprit, amyloid beta plaques. Even after billions of dollars in research, removing amyloid plaques often didn't restore memory. So the old way of thinking about it was that memory was stored in synapses in the synaptic network, and when these plaques form, it destroyed the connections and memory was lost. But now we're thinking about memory as a more distributed property in living organisms where there's hidden networks that play a role in memory that are made up even inside cells. So it's not just the networks of the cells themselves, but it's the hidden networks inside the cells. So we'll talk about that more in future slides. And if you would want to read more about the role that this hidden mechanism plays in evolution, even since the dawn of life, you can check out my paper titled Liquid-Liquid Phase Separation as a Driver of Abiogenesis and Evolution. And it's in academia, molecular biology, and genomics, open access, so everybody can see it. So, the classical view of memory. For most of the 20th century, memory was thought to be stored primarily in synapses, the connection between neurons. This idea emerged from the famous principle neurons that fire together wire together. This model explained many observations about learning and memory, but it also created a simplified narrative. Memory loss occurs when neurons die or synapses break down. When researchers discovered large deposits of amyloid beta plaques in the brain of Alzheimer's patients, the explanation seemed obvious. Amyloid plaques were assumed to damage neurons and erase the synaptic patterns that encode memory. So, when these nerves fire together, the ones that fire together on top right here are the ones that start to wire more connections together. So that is where the saying comes from, neurons that fire together wire together. And then this picture to the left shows amyloid beta plaques in red. And when those form, they're going to inhibit these connections from forming. And this was the old way of looking at it. So the amyloid hypothesis, to go deeper in the classical view, for more than 30 years, the amyloid hypothesis dominated Alzheimer's research. The idea was straightforward. Amyloid beta accumulates in the brain. These plaques disrupt neuronal communication. Synapses degenerate. Memory is lost. This theory guided thousands of studies and dozens of clinical trials, but cracks slowly began to appear. Some individuals had large amounts of amyloid plaques but no cognitive impairment, while others experienced severe memory loss with relatively little amyloid accumulation. Many drugs that successfully removed amyloid plaques showed minimal improvement in memory. This suggested something profound. Amyloid plaques might be part of the story, but not the whole story. So this is a schematic to the left showing that amyloid plaques gets disrupted, it gets misfolded, and then it oligomerizes or combines together with itself through self-assembly into these amyloid fibrils that accumulate in the brain. And this is something I talked about in previous videos and in my paper called Liquid-Liquid Phase Separation, which is thought to be a normal function of amyloid beta plaques. So it normally makes this dynamic scaffolding with water, but when this happens, it probably sequesters itself with very little water, forming very solid, dense fibrils, and then that is the reason why these plaques form. So these are intrinsically disordered proteins, so normally it's partly intrinsically disordered, normally very dynamic and can form a dynamic scaffolding, but it gets misfolded and ends up oligomerizing or self-associating with itself in a more dense structure, which ends up making and forming these plaques. So memory is not localized. Modern neuroscience increasingly supports a different perspective. Memory is not simply stored in individual synapses or specific neurons, but instead it appears to be distributed across multiple biological scales. Memory involves synaptic protein networks, cytoskeletal structures, RNA regulation, epigenetic modifications, metabolic states, large-scale neuronal network dynamics. In other words, memory may be an emergent property of interacting biological systems rather than a static structure stored in one location. This shift has opened the door to an entirely new mechanism that may organize memory-related molecules inside cells, a mechanism known as liquid-liquid phase separation. So this is an example of liquid-liquid phase separation. And you can see that under certain conditions, these two different bits of information, red or green, will organize themselves with each other into two different layers. And this is an interesting mechanism for memory because you can think about it being delocalized and spread out. And then when one bit of information, whether it be something you hear or something you think about or something you smell, that one bit of information can start to initiate this organization to occur and this memory to form. So that makes the system a lot more robust since it's delocalized and can be accessed from many different perspectives. So let's talk about liquid-liquid phase separation. And these are two figures from my paper that I told you about on the first slide. Inside cells, many proteins and RNA molecules don't simply float randomly. Instead, they can self-organize into liquid-like droplets. This process is called liquid-liquid phase separation, or LLPS. If this sounds unfamiliar, think about oil separating from water. Two liquids can spontaneously organize into distinct phases without membranes. Cells use this principle to create dynamic molecular compartments. These droplets can concentrate specific proteins, organize biochemical reactions, regulate gene expression, and coordinate signaling pathways. And I talk about all this in my paper, and since it can organize things without a membrane, that's why it's thought to play a role in the origin of life. Importantly, many proteins involved in synaptic plasticity and RNA regulation are capable of phase separation. This means memory-related molecules may assemble in temporary biochemical hubs inside neurons. So this is just showing a easy to understand depiction of oil and water, which is a classic example of liquid-liquid phase separation. But when you think of it happening in biological cells, it's more complex. So you can think about it in a sigmoidal curve and how these individual oil droplets composed of RNA and proteins can create a network of on and off switches. So when you don't have enough molecules, the concentration isn't high enough to create this liquid-liquid phase separation, it would be turned off. But then when the concentration of proteins and RNA or post-translational modifications or maybe ions or any organic substance that's inside the cell or outside the cell, when you have enough concentration of that, then all of a sudden it gets turned on and you have condensation. So that's how this mechanism can create a network of on and off switches, which can act as a circuit of memory in the brain. And not only in the whole brain, but even inside cells and organelles within those cells. So phase separation in memory. Recent research suggests that synaptic proteins can form phase-separated condensates, which is the liquid-liquid phase separation we talked about previously. These condensates act like molecular organizers, bringing together the components needed for learning and memory. For example, proteins in the postsynaptic density, the dense molecular region where synapses receive signals can form dynamic protein clusters through phase separation. These clusters can stabilize synaptic changes, regulate neurotransmitter receptors, and organize signaling pathways that strengthen synapses. In this framework, memory isn't just stored in a connection between neurons. It may also depend on the physical state of molecular assemblies inside those neurons, and it can happen outside the neurons. Memory becomes something dynamic, fluid, and distributed, and that is what makes it robust. That's why you can hit your head very hard and you'll still maintain memory, although not too many times. And disruptions in phase separation may contribute to the neurodegenerative diseases. A new view of neurodegeneration. I just need to take a sip real quick. In this newer view, amyloid plaques may not be the original cause of memory loss. Instead, they might represent a downstream consequence of disrupted cellular organization. Many neurodegenerative disease proteins, which are shown here, are known to undergo phase separation. Under healthy condition, these proteins form reversible liquid droplets. But under stress or aging, these droplets can solidify into pathological aggregates. This suggests that diseases like Alzheimer's might involve a failure of dynamic molecular organization that normally supports memory. So the new view, which I've shown depicted here, is that memory isn't just stored in neurons. It emerges from the coordinated organization of neural networks, synaptic proteins, dynamic molecular droplets formed through liquid-liquid phase separation. So it's an emergent phenomenon at many different scales which form memory. So in conclusion, the story of memory is evolving. Instead of a single protein destroying memories, we're beginning to see memory as something far more distributed and dynamic. Process shaped by networks of neurons, molecular interactions, and physical principles like phase separation. In this view, memory isn't stored in one place. It emerges from the organized complexity of living systems, and understanding the complexity may be the key to protecting memory in the future. And this complexity can actually be explained through complex adaptive systems, which I think explains not only organisms, but ecosystems and evolution itself. So I have a schematic here just showing going over things that we talked about previously. And also, if you want to learn more about complex adaptive systems, you can read my paper, Chaos to Coherence, A Path to Molecular Organization, which is published in the Taylor and Francis Group first edition book, Quantum Mechanics of Biological Systems. So the take home is the next time you remember a childhood moment, you might not just be recalling a pattern of neurons, you might be witnessing the coordinated dance of molecules, droplets, and networks that make memory possible. This mechanism has been unraveling for billions of years on Earth in all living organisms since the dawn of life. Check out my paper, Liquid-Liquid Phase Separation as a Driver of Abiogenesis, aka The Origin of Life and Evolution, if you would like to read more about how this mechanism plays a role in the dawn of life and evolution of signaling pathways on Earth. I hope you enjoyed the video and you have a wonderful day.